Hello everyone, my name is Madhura Nayak and I'm a product manager at ACD. Thank you for joining our session today, and we are very excited to talk about our newest product, the RNA protein co-detection ancillary kit, which enables compatibility of your choice of IHC antibody with key RNAscope and BaseScope assays. With that, I would like to go over the agenda for today, where in the first part, I will briefly touch on our technology play in spatial transcriptomics field and touch on the key applications enabled with dual detection and how the multiomics approach is needed to further our understanding of unique biological questions. And then I will introduce you to our new product offering. Okay, so where exactly do our products fit in the research space? As we all know, complex, highly heterogeneous tissues such as the brain and tumors, as well as processes such as embryonic development, are comprised of multiple cell types and states. And as technology enables, researchers are interested in characterizing transcriptional activity more fully. These efforts can enable identification of new cell types, predictive biomarkers, and new therapeutic strategies, and more. These goals require techniques that provide highly sensitive, specific, and multiplex spatial approaches with single-cell resolution. The RNAscope technology provides all these, so it's being applied to these questions quite regularly. With that, I would like to segue into three unique assays that we have at Bio-Techne to detect RNA in the tissue context at single molecule sensitivity with single cell resolution. The RNAscope assays for mRNA and non-coding RNA greater than 300 nucleotides in length, and BaseScope for exon junctions, splice variants, and highly homologous or short sequences. And our newest assay, the microRNAscope, which can detect even shorter RNAs and support ASO and siRNA detection in addition to miRNAs. Before we introduce you to the product, I wanted to touch briefly on the research need. Fundamentally, researchers are trying to understand the mechanism of normal state versus disease state. And this is how well the cell interactions, functions, etc. are characterized in a tissue context. Such levels of characterization is not possible with a single analyte assay, and hence the need for combination. Additionally, each assay comes with its own limitation. IHC utilizes antibodies, which for popular targets are well established. However, for some targets may lack the desired specificity and sensitivity, and this causes the need to correlate with RNA detection method. This brings the need for an assay that can combine both analyte detection with morphological context. And with that, I would like to introduce you to our new reagents that enable these applications. The new reagents in the form of the RNA protein co-detection ancillary kit, when used with the new workflow, will allow researchers to simultaneously examine cell type specific gene expression and identify cellular sources of secreted proteins. Some of you may have already tested certain antibodies and found them incompatible with our RNAscope and BaseScope assays. Our aim with this new assay is to enable more antibodies to be compatible with our assays, and we will see later some examples of key antibodies that were previously incompatible are now compatible with the new workflow. The new assay essentially contains three parts: the new RNA protein co-detection ancillary kit, which includes the co-detection blocker, co-detection antibody diluent, and the co-detection target retrieval reagents. This kit has been validated with our chromogenic red and multiplex fluorescent assays on the manual and Leica platforms, and also has been validated with our BaseScope assays. Along with this kit, we would also like to introduce you to the new protocol where the key primary antibody step is now performed right after the target retrieval step, following which we go on to perform the rest of the RNA-H steps. We then follow this up with secondary antibody to detect the protein of interest on the same section. This workflow has been designed to be completed within 2 days, with the primary antibody incubation lasting overnight as a breakpoint step. So with this new integrated workflow approach, we can now enable more antibodies to be compatible with the RNA protein co-detection, and reduce the need for extensive screening for compatible antibody clones, which results in faster time to results. For those of you who may have tested the sequential workflow before, this new workflow and reagents have supported equivalent or better performance of antibodies. This also supports equivalent sensitivity to ISH-only detection and additionally more faithfully retains specificity of antibodies, so which is IHC alone. In this slide, I'm showing what a signal from an IHC-only run looks like in the left panel. And when we add the RNA-scope pretreatment, we lose that signal as shown in the middle panel here. And then in the final panel on the right, with our integrated workflow and new pretreatment reagents, we can now rescue the IHC signal. And this, in a sense, makes it easier to combine your antibody of choice with RNA-ish. An example is the CD8 antibody shown here in the left panel with the standard IHC, in the middle panel with ISH and IHC without the new reagents and protocol, where we lose the signal. And in the right panel, we are now able to visualize both signals with the new co-detection reagents. Now I would like to take you through some examples of staining generated with the new reagents in some of our validated assays with the IHC antibodies. In the first example here, we show some data with CD4, CD5, and FOXP3. CD markers are cell surface markers which are very useful for identification and characterization of leukocytes and different subpopulations of leukocytes. FOXP3 is a protein involved in immune responses and known to be a master regulator of the regulatory pathway in the development and function of regulatory T cells. These represent fairly standard proteins utilized in oncology research and shown here combined with RNA-ish can be used to get more data out of your precious samples. The samples shown here are tonsil, head and neck cancer, and as you can see from the panel on the right, both RNA-ish and IHC signal is maintained with the new workflow and reagent with a Leica red assay. Another example of staining here, the protein detection is performed using a green chromogen, giving a great contrast on tissues where an alternate to the brown DAB is preferred, perhaps due to tissue artifacts. Shown here are examples of CD3, CD8, and Ki67 antibodies in the left panel as control IHC alone, in the middle panel combined with RNA-ish without the new reagents and workflow, and in the right panel we observed optimal signal detection with both the RNA and protein with the new workflow and the new reagents. And here we show compatibility of these reagents and workflow with the CD68, CD20, and Ki67 antibodies and our manual base-scope assay. This is an example of 3-plex-ish with Ki67 antibody with this new reagents and workflow, a key example of how you can get much more data with more targets and detect both RNA and protein on the same slide. And lastly, here are some more examples of CD markers detected with the brown or the green chromogen along with the red-ish signal. And with all that data, I would now like to conclude and summarize today's presentation. In today's session, we have shown some key examples of how multiomics profiling with single-cell resolution can be achieved with the new reagents and workflow. With this new ancillary kit, you can now incorporate RNA-ish into your existing IHC workflow and gain deeper insights and preserve precious samples. This reagent and workflow have been extensively validated with the red chromogenic multiplex fluorescent assays on our manual and Leica platform. And lastly, we hope that with this product and key research examples we have shown in this deck, you're now able to utilize the power of two technologies, IHC and RNA-ish, to answer your research questions. And with that, I would like to thank you for your time and attention today. Thank you.